Okay, so we're at the stage where we're getting ready to load the library for the flow cell, and the very first thing, given the concentration that I have, is I'm going to need to, I need a total of 12 microliters, and so I have to dilute everything. So I'm going to call this my final library, or loading library. How about that? $12 is what I need, so let's dilute this, adding 1.5 like microliters, 10, 1.5, and 1.5 of my really concentrated library. Okay. Oh, that is the loading library. I'm getting close to the 300. So now what we need to do is prepare the loading mix for the flow cell. So we're going to have a mix of flow cell flush, BSA, and flow cell tether. And we've already done the flow cell check. So what I'm going to do is I'm going to prepare this, and I'm going to change the setup just a little bit so that we can get pretty good close-ups, because I need to prime the flow cell and I need to then actually load the library. So let's go ahead and make the flow cell flush mix. That is 1170 of the flow cell flush. Already thawed. Five microliters of BSA. This is one of the items that's not included, so you'll need to purchase it. But we don't use much, and I guess it goes a long way, so good to keep a tube around. Might be worth pre-aliquoting, because it's, I always like to avoid freeze thaw, and we use so little of it. Okay, 30 of the tether. This is really important. It gets the keeps the complex of the DNA and motor proteins and all those things proximal to the nanopore, and increases dramatically, because I know that, I guess, because I've forgotten it in the past. Okay, I've made this flow cell flush And I'm going to change my setup so that we can get nice close-ups of the loading procedure. Before that, I guess I should say this is our MinION flow cell. I've opened it. I've already done a port check. I always like to label these on the back with the date, verify that I really know the format of the cell, all the details. But when you open it, what you get is the flow cell in a little case. It's got a little bit of absorbent. There's a light shield, this piece of plastic that is there. Don't throw that away, you're gonna use it. A little absorbent tray. But let me change my setup so that we can see it, because this is the disposable flow cell itself. And then we also have the MinION M1KD, brand new, first time using it for me, that we are going to load. And so I want to make sure you get close-ups of this. So give me a moment to reset. Okay, so we are going to go ahead and get started. I've got the device here, and I'm going to go ahead and lift the cover. And what you should see, I'm actually going to turn this around so that it's facing the proper way. There are the places where the electronics plugs will go. This element, I think, probably is also help there to regulate the temperature. So let me center that. And I'm going to take a flow cell, the flow cell, my only flow cell. And I want to push it forward. I don't want to crush those pins. Actually, it's worth showing that underneath there are these electronics that we don't really want to touch or damage. So I'm going to push it forward, and it will naturally sort of click, and then I can easily press down those pins without any resistance. There are a couple of different ports because liquid needs to move around, and air and various things. So this port we don't need to touch. That's going to be where we do the loading in a bit. But for right now, I need to swing open the priming port. There's a hole there. There are also other holes that allow liquid to flow, but right now, because I have that priming port open, I'm going to be able to get some liquid out. Essentially, there is an air bubble that prevents liquid from flowing in the wrong direction while this is sitting in any, I don't know, any manner of orientations during shipping. But what I'm going to do is make sure that we have a complete channel or a contact so that I can move the liquid as I add things and the fluidics can work. This, I will say, is the most dangerous part because if we're not careful, air can flow into the system, and if air touches that area here, that little yellow window is really where the nanopores are, it will irreparably damage. So following the instructions, I'm taking a P1000 pipette and setting it to 200. And then what I want to do is I want to get this pipette directly into, it fits nicely, press it with a little bit of a seal into that priming port. And parts of this is going to be hard to see, but I'm going to try to get down there and focus as best I can. Let me adjust this. So I've got that tip there. And then what I'm going to do, I'm quite zoomed in, but hopefully with enough detail, resolution doesn't look too bad. I'm going to keep it there. And what I'm going to do, maintaining a seal, is I'm going to slowly turn the dial on this out of focus pipette. But you can kind of see I'm going to turn this dial gently, and I don't want to. I'm going to increase the volume from 200 to 230, 220, 230, and I'm doing it slowly. And the point is that I'm going to start doing it now. As I start doing it, I'm creating a vacuum, but I don't want it to go really, really fast, because what will happen is that for the first several microliters, I'm at 217, nothing will appear to be moving in the system. And what I'm looking for is a little bit of liquid to come into the pipette. but it doesn't seem like it's working. But theoretically, if I keep going and increasing that pressure, I'm almost up. I'm almost at 220, I'm at 229, at 230, and at 230, going slowly enough, you might just barely be able to see down there that I've started to get a little bit of liquid into the system, and that's all that I want. That 30 microliters of liquid is enough to create a continuous fluid throughout the system. That's all that I'm looking for. So I can stop there. Now, I can change tips. I've drawn up just a little bit of buffer. It's not that hard, but it does have to be done slowly and with patience, and you can build your confidence to see someone do it. Now, I'm gonna load 800 microliters and wait for 5 minutes. So I'm resetting my pipette, as you may be able to hear, to 800. And I'm taking up 800, roughly half of this flow cell flush. I've just mixed it pretty thoroughly. And what I want to do now, when I go in to add that, I am going to tune this down so slightly, just to ensure there's like a little bit of liquid on the tip there, because I don't want to introduce any air bubbles. So I want to make sure there is some liquid down there. And now I'm going to slowly lower the volume of the pipette, and let me zoom out just a little bit. What you may be able to see is that as I do this, liquid is going to start flowing in a serpentine fashion through these channels down here. Let me see if I can focus a little bit. Yeah, liquid is going to start moving. And I could do this slowly. I've taken a long time to get here, so in my opinion, I'm gonna have to go molasses, but I can go slowly. I think to whatever extent going slowly, can help minimize that we're replacing liquid in the entire system, fluidics in the entire system, and those nanopores are just fragile proteins. So just a few microliters at a time, going slowly, I hope will minimize the chance of damage. We're coming up almost on 1 a.m. here, folks. What a wonderful time to do nanopore sequencing. So if you've stuck with us, this is just the very last little bit. Okay, I'm gonna stop before I've actually gotten all that liquid out. And the last few drops, there's enough of a flow in the system that I can drop those last couple drops and it absorbs. I don't have to get every last drop. 5-minute timer, and then we will resume. Okay, so while we are waiting 5 minutes to be able to continue the loading, I can now prepare the final loading mix, which is going to be sequencing buffer, library beads, and the library, the 12 microliters of the final library. Okay, and to that, we need 37.5 microliters of the sequencing buffer. everything in order as we have been doing all along. 25.5 of loading beads. These are tiny little glass beads. They need to be agitated, stirred, vortex. Library beads. Shaking them works. You really want to make sure that you don't have a pile of them. This is looking really good to me. 25.5. So you want to use it immediately or get them out immediately. We will have to do this again. But it should be quite uniformly opaque, milky solution. And these help to disperse the library across the surface of the nanopore, the nanopores. And then 12 microliters of the library. This all takes time, but it's not hard. It just takes time. Okay, this is what we are going to load. Got 2 minutes here. So we'll have to shake this or stir this a bit. We are going to load another 200 of this flow cell flush priming mix onto this into the system once we reach 5 minutes, and then we're gonna mix this, add this, and then we are ready to run it. So 1 minute and 40 seconds. Okay, it is time. So I am going to go ahead and add another 200 of the flow cell flush. That is relatively easy now that we have done the priming of the flow cell. So it's just another dose of that liquid. Flow cell priming mix, 200. Same tips. Get a little drop of liquid at the bottom before you make contact to make sure that you're not adding any air into the system, and slowly turning that down. You will see the liquid. I can do this with one hand or try to make sure that you see that liquid moving down that waste channel. And stopping before I get to the last drop, I can let it go without. Okay, so now I've got 75 microliters of library to add, and the way that I do that is now we draw our attention to this port. There's a little plug and it actually pulls up. We don't want to rip the tape, so we just pull the plug out and put it to the side. Okay, so this is essentially where everything, all the action takes place. The nanopores are on that little yellow window. I'm quite zoomed in here, so I'm doing my best not to make it too blurry. But that is where we're going to, in a dropwise fashion, add the library. I'm gonna pipette up and down just a couple times to make sure everything is really loaded and those beads are really nicely suspended. But this is pretty easy because I need to just give a drop and it will flow in one drop at a time, and you may be able to just see those beads just sort of disperse. Missed the drop there. It's all right, suck it back up. Okay, could have done that a little bit more carefully. It's a little bit more difficult at that angle, but it makes for good filming. I'll replace this plug minimize evaporation, but I also got to place that there. I will take the light shield, which they learned exposing this to light harms the process a bit. So this fits over it to keep it nice and dark. You can close the port, and this is ready to go ahead on to the, well, to be connected to the computer. So that's it. We'll see how the sequencing goes, but that is the entire preparation of a library using the native barcoding kit, in this case with genomic bacterial DNA. So time to run it on the computer using the MinION software.